functional in the first step.
Oh, no.
I get all my letters in there, which is filtration. Okay. After that, in the proximal or distal convoluted tubule, we're going to be doing reabsorption and secretion. In the proximal convoluted tubule, primarily, the reabsorption and secretion is not regulated. It's determined by how much stuff there is. So if we have a lot of sodium, we reabsorb a lot of sodium. If we have a lot of glucose, we reabsorb a lot of glucose. If we have a lot of hydrogen ions in the blood, we secrete a lot of hydrogen ions into the filtrate. When we get to the distal end of the tubule, it's more going to be regulated. So about 80%, 85% of our sodium ions have already been reabsorbed. Now it's dependent on how much more water, blood volume the bodies need. So the distal tubules, and we'll see this on Thursday, then the body will have aldosterone. Will bind to cells of the distal tubule. Will increase the number of sodium-potassium channels. All right, so that part is regulated. But we're going to look today just at the proximal convoluted tubule. So the proximal convoluted tubule is made up of simple cuboidal epithelial cells. And these simple cuboidal epithelial cells, and all of them look like this, not just the ones I'm drawing on, have these long extensions. Are these microvilli or cilia? They look like cilia because of their length. Are cilia modal or non-modal?
Modal.
Modal. We saw them in the respiratory pathways where they swept mucus. We saw them in the oviducts where they swept along the egg. There's nothing to sweep along here. What's more important, surface area or sweeping the filtrate?
Surface area.
Surface area. So even though these are very long, they are actually microvilli. Okay? So if we move back to this previous histology slide that you looked at the macula densa on, the film looks like it's solid, but these cells here, these are actually very long microvilli. The surface of the cell is about right here. So these are all very long microvilli. It creates a messy look to the inside of the lumen.
These are simple cuboidal?
These are simple cuboidal, yes. All right, so typically their nucleus is in the upper half of the cell. And we have a simple cuboidal epithelium. For reabsorption, an increase in surface area for reabsorption and secretion. So, as we look at that process, compare those cells with these cells. These are distal convoluted tubule. Hardly any. They're very short and sparse. All right?
Where was the proximal one?
The proximal one is proximal to the capsule.
No, I mean on the...
Oh, on this one?
Yeah.
These would be proximal. Here, here, here. They look like they have a thicker wall and they have this irregular appearance to the microvilli, those long microvilli. Another view of proximal epithelia would be in this image here. This is a lower power. So I'll see if you guys can pick out. The circle is surrounding the capsule. Can you find the proximal convoluted tubules?
Those up there.
The messy ones?
There.
Here,
There.
Over there.
here,
Over to the right.
all of these, down here. Those are the proximal convoluted tubules.
So it's the
Okay.
microvilli that makes them look thick. Huh?
Yes. Yeah.
Good.
And they have a lot of mitochondria, so then they're staining more pink as well, because that causes them to stain a deeper pink. This would be a distal tubule right here. And this would be a collecting duct.
Because you can see more of the cells versus.
Right. Now, we don't see the plasma membrane separating these cells, because the plasma membrane is interdigitating. So I really don't need that capsular diagram anymore. You guys okay if I erase it? Okay. So I'm going to diagram a couple of cells of the proximal convoluted tubule. I will eventually get to this slide on the board. I haven't forgotten it. This will be filtrate side. I suppose I should draw it with a yellow marker, but that doesn't show up very well. And these cells have very long microvilli. And their lateral walls and their basal walls have infoldings, but not thrown into folds. All right? So these would be the microvilli. And these are identified as basal lateral foldings.
So this is one cuboidal cell, right?
So far, yes.
So far.
Here's another one. And there are tight junctions between them. These are loose tight junctions. Remember the tight junctions that the brain capillaries had? These are loose tight junctions. Water molecules, chloride ions can still pass between them. And the plasma membrane is infolded and it makes it difficult to see in a light microscope. It's hard to see the actual membranes between the cells. So this is called the apical surface. And this is called the basolateral surface. And then we have our peritubular capillary blood. Blood. And this would be the proximal convoluted tubule. Okay. Now, remember what I said we have in the filtrate? If you're not sure, you can look back on page 12. I believe it's 12.
Proximal convoluted
So down on the
tubule.
bottom of page 12. You see all those ions and contents, all of that had to be filtered. All right, so we're going to have in our filtrate, we're going to see sodium ions. We're certainly going to have chloride ions, because that's what we find with sodium. Calcium, magnesium, ammonia, which we can form into ammonia ions. Phosphate. We also have molecules such as glucose, individual amino acids, urea, water. So all of those components were filtered into the glomerular filtrate. Okay. And now we're going to take a lot of it, most of it, back into the blood. Okay.
It's GFR, right?
Yeah. GFR is just glomerular filtration rate. So sodium is in higher concentration outside the cell or inside the cell?
Outside.
Outside the cell. So if we had a sodium ion channel, it would leak into the cell. How would we get it out of the cell into the blood? With a sodium potassium or sodium hydrogen pump. Okay. So if we put that sodium potassium pump in, we're going to put it here. So this would be our sodium potassium pump, also known as an ATPase pump, because it requires an enzyme that breaks down ATP to provide it the energy to move it up the gradient. So sodium is moved out of the cell against its gradient, and potassium is moved into the cell against its gradient. And so is this identified as, we know it's active transport. Is it an antiport? Is the pump an antiport or a symport?
Anti, it's going.
It's an antiport because they're going in opposite directions. Okay? So this is active transport via an antiport. Now, given that sodium ions are going to diffuse into the cell, all right, using this active transport is going to move those sodium ions out, and it's going to keep that sink low. It's going to keep the numbers of ions inside the cell low. So let's turn on the pump. We have fewer and fewer and fewer sodium ions in the cell. So now at the other end of the cell, the apical end, next to the filtrate, we have another transport protein, but this one is involving what's called secondary active transport. All right? So in this case, this protein has a channel for sodium. It also has a channel for glucose or amino acid. Which has the higher drive to go in the cell, the sodium or the glucose?
Into the cell?
Into the cell.
With charge-
Both want to go in.
Would the charge be sodium?
So the sodium has the higher drive. What have we done when we turned on the pump? We increased that drive. So by having a molecule that transports two different components, one which has a high drive to enter the cell, we have coupled that passive process to an active process. And that is described as secondary active transport. So sodium ions are moved into the cell, and glucose is moved into the cell by the gradient driven by the sodium potassium pump. So it is a passive process. This itself does not require energy, but it works because of the pump, which does.
We get the sodium going in, because the sodium's leaving. Why is glucose going in?
Because glucose diffuses in, it normally requires a facilitated diffusion protein. It happens because of this transport protein. In this case, it's using amino acids, but it could use glucose as well. Both of them have sites that fit on the, so normally with glucose, we have to flip it over. So it's called a facilitated diffusion. We don't use energy to flip that over. But sodium, if all we had was just sodium, it's going to move through this really fast. Okay? By coupling it and having a site there for glucose as well, it pulls the glucose in with it. If we had a separate protein that would take the glucose, that would move in much more slowly than the sodium. The sodium is moving in fast because we've depleted the sodium by our pump. So by using the same protein that fits both sodium and glucose,